Hello everyone, Bill Nye here, and today, we're about to embark on an incredible journey into the microscopic world that shapes who we are. Imagine a universe so small, it fits inside every cell of your body, yet it holds the secrets to life itself. DNA is the instruction book inside almost every cell in your body, coding for everything from your eye color to how you fight off germs. It's a vast, complex manual, written in a language of 4 simple letters, A, T, C, and G, that together spell out the blueprint for every living thing on Earth. Now imagine if we could edit that book, change a word, a sentence, or even a whole chapter. What if we could correct genetic typos that cause disease, or even add new instructions to help us adapt to our environment? This is no longer science fiction. It's real, thanks to a revolutionary technology called CRISPR-Cas9. In just a few short years, CRISPR has transformed from a scientific curiosity into a powerful tool that's changing the way we think about biology. CRISPR is like a pair of molecular scissors, able to cut DNA at a precise spot. Scientists can use it to snip out faulty genes or insert new ones, with a level of accuracy that was unimaginable just a decade ago. It's revolutionizing medicine, agriculture, and biology. Doctors are exploring ways to treat genetic diseases at their source. Farmers are developing crops that can withstand harsh climates, and researchers are unlocking mysteries of life that have puzzled us for centuries. But what exactly is CRISPR? The name stands for Clustered Regularly Interspaced Short Palindromic Repeats. A bit of a mouthful, but it's actually a clever system borrowed from bacteria, which use it as a defense mechanism against viruses. We'll break down what that means, where CRISPR came from, and why it's so exciting. We'll see how scientists discovered this hidden code in bacteria and figured out how to harness it for our own use. We'll trace its journey from a strange pattern in bacterial DNA to a world-changing technology. Along the way, we'll explore the challenges, the breakthroughs, and the ethical questions that come with editing the very code of life. By the end, you'll understand why CRISPR is a monumental leap forward in science, a tool that could help cure diseases, feed the world, and maybe even change what it means to be human. So let's get started. The world of genetics is waiting, and the possibilities are endless. To truly understand CRISPR, let's shrink down to the microscopic world of bacteria, a realm teeming with life, invisible to the naked eye, yet fundamental to all ecosystems on Earth. Here, in this hidden universe, bacteria are constantly at work, multiplying, evolving, and surviving in environments as diverse as the human gut, deep ocean vents, and even the icy poles. But these tiny organisms are not alone. They face relentless attacks from viruses known as bacteriophages, or simply phages. These phages are specialized predators, each evolved to target specific bacteria, and they outnumber bacteria by a factor of 10 to 1 in some environments. When a phage finds its bacterial target, it attaches and injects its own genetic material, DNA or RNA, into the cell. This hijacks the bacterium's machinery, forcing it to become a virus factory, churning out new phages until the cell bursts, releasing a fresh wave of invaders. For billions of years, bacteria and phages have been locked in a high-stakes evolutionary arms race. Each side constantly develops new strategies to outwit the other, driving innovation at the genetic level and shaping the course of life on Earth. One of bacteria's most ingenious defenses is CRISPR, short for Clustered Regularly Interspaced Short Palindromic Repeats. This adaptive immune system allows bacteria to recognize and remember past viral invaders, giving them a powerful edge in the ongoing battle for survival. When a bacterium survives a viral attack, it doesn't just move on. Instead, it snips out a small fragment of the invader's DNA and carefully inserts it into its own genome, in a special region called the CRISPR array. This process is like collecting a mugshot of the enemy for future reference. The CRISPR array acts as a genetic scrapbook, a living archive of past viral encounters. Each entry in this archive is a record of a specific virus that the bacterium or its ancestors have survived. Each stored snippet, called a spacer, is a unique fingerprint of a virus. These spacers serve as molecular mugshots, allowing the bacterium to quickly recognize and destroy the same virus if it attacks again. What's even more remarkable is that these spacers are passed down to future generations, giving inherited immunity. This means that the descendants of a bacterium are born with a ready-made defense system, already primed to fight off familiar threats. The CRISPR array is a living record of battles fought and won, written directly into the DNA of bacteria. It's a testament to the resilience and adaptability of life at the smallest scale, This ancient microbial defense system is the very foundation of the revolutionary gene editing tool we use today, CRISPR-Cas9. By harnessing nature's own strategies, scientists have unlocked new possibilities for medicine, agriculture, and beyond. Nature's solution to survival has become our tool for innovation, allowing us to edit genes with unprecedented precision and speed. What began as a microscopic battle for survival now shapes the future of science and humanity. And all of this, these epic struggles, ingenious defenses, and groundbreaking discoveries, happens invisibly all around us every single day. So how does a bacterium use its viral DNA library to defend itself? Imagine a tiny cell constantly under threat from invading viruses, yet equipped with a sophisticated memory system. This system is like a living memory card, storing fragments of viral DNA as a record of past infections. When danger returns, the bacterium is ready. Enter the CA's proteins, CRISPR-associated proteins, the workhorses of this immune system. These specialized molecular machines are always on alert, waiting for the signal to spring into action. The bacterium transcribes its spacer DNA into guide RNA molecules. These guide RNAs are like personalized wanted posters, each one designed to recognize a specific viral intruder. Each guide RNA carries the code of a specific enemy virus. It's a molecular memory, a record of past battles ready to be used if the same virus dares to attack again. This guide RNA teams up with a CA's protein, most famously Cas9. Together they form a search and destroy unit, patrolling the cell for any sign of the invader. Together they patrol the cell, searching for matching viral DNA. The guide RNA acts as a homing beacon, leading Cas9 directly to the target. If a familiar virus attacks again, the guide RNA locks onto the invader's DNA. This recognition is incredibly precise, ensuring only the enemy is targeted. Cas9 then acts as molecular scissors, making a precise cut in the viral DNA. This cut is not random, it's targeted, efficient, and devastating for the virus. This cut disables the virus, stopping it from replicating. The threat is neutralized before it can cause harm, and the bacterium survives another day. It's a precise, elegant defense. Remember the enemy, find the enemy, cut its DNA. This cycle of memory and response is what makes the CRISPR system so powerful. This natural system is the blueprint for today's gene editing revolution. Scientists have learned to harness this ancient bacterial defense for our own purposes, opening up new possibilities in medicine and biotechnology. Nature's ingenuity, now in our hands. What began as a simple survival strategy in bacteria has become a transformative tool for humanity. The guide RNA and Cas9 are the ultimate security team. Their partnership is a testament to the power of evolution and adaptation. With this, bacteria survive, and so can we, by harnessing this tool. Isn't nature amazing? The story of CRISPR is a reminder that even the smallest organisms can teach us the biggest lessons. For years, scientists saw strange repeating sequences in bacterial DNA but didn't know their purpose. The breakthrough came when researchers realized these spacers matched viral DNA, evidence of an adaptive immune system. Food scientists working with yogurt cultures proved bacteria could acquire new spacers and gain resistance to future infections. The next leap was understanding how the system worked. CA's proteins cut DNA, guided by RNA transcribed from the CRISPR array. Scientists wondered, if bacteria can program a protein to cut viral DNA, could we reprogram it to cut any DNA we want? The answer was yes. This was the moment CRISPR became a revolutionary tool for all of biology. A new era of gene editing had begun. The leap from bacterial defense to gene editing tool was made by Emmanuel Charpentier and Jennifer Doudna, two visionary scientists. Charpentier discovered that two RNA molecules, crRNA and tracrRNA, work together to guide Cas9. Teaming up, she and Doudna engineered a single guide RNA, making the system simple and programmable. Now scientists could design a guide RNA for any gene, pair it with Cas9 and cut DNA wherever they wanted. Their 2012 paper showed this system could edit DNA at will. For this, they won the 2020 Nobel Prize in Chemistry. Their work made gene editing accessible to labs worldwide. This wasn't just an improvement, it was a revolution. Science would never be the same. Before CRISPR, gene editing was possible, but slow, complex, and expensive. Two main tools were zinc finger nucleases, ZFNs, and TALENs. Both required building a new protein for every DNA target, a major bottleneck. ZFNs used engineered zinc fingers to recognize DNA, but designing them was difficult and costly. Talons improved on this, using repeating units to target DNA but still required custom proteins for each gene. Both methods allowed targeted gene edits but only in specialized labs. They proved gene editing was possible but not practical for most scientists. The field needed something faster, simpler, and more accessible. CRISPR was the answer. It democratized gene editing, opening the door for rapid innovation. The stage was set for a revolution. Science was ready for a new tool. CRISPR-Cas9 changed everything by making gene editing simple and accessible. Unlike older tools, you don't need a new protein for each target, just a new guide RNA. Making guide RNA is fast, cheap, and easy. Now almost any lab can do gene editing in days, not months. This has accelerated discovery across biology. CRISPR is also more accurate and efficient, with fewer off-target effects. Its reliability makes experiments easier and results clearer. The same Cas9 and guide RNA system works in bacteria, plants, animals, even humans. This versatility has opened new frontiers in medicine and agriculture. Scientists are creating disease-resistant crops and exploring cures for genetic diseases. CRISPR isn't just an upgrade, it's a platform for innovation. The rules of biology have changed. The revolution is here. Let's recap our journey. We explored gene editing and discovered CRISPR-Cas9, a tool borrowed from bacteria's ancient immune system. We saw how bacteria store viral DNA as a genetic memory, and how scientists turned this into a programmable tool. Charpentier and Doudna's insight made gene editing simple and accessible. Compared to older tools, CRISPR is faster, cheaper, and more accurate. It's fueling an explosion of research and discovery. The code of life is now editable by anyone with the right tools. The future of biology is wide open. We've covered what CRISPR is, where it came from, and why it's revolutionary, but there's more. After CRISPR cuts DNA, the cell's repair machinery takes over. Scientists can harness these repair processes to disable genes or insert new genetic information. Next time, we'll dive into how cells repair DNA and how this lets us rewrite the code of life. The ability to edit genomes is just beginning to reveal its full potential. Medicine, agriculture, and science are being transformed. The future is being written and rewritten right now.